All right, so good morning, good afternoon, everyone. Welcome in from across the continent and beyond. We've got folks here in Alberta, Wisconsin, California, Ontario, and more. A big welcome to all of you for this exciting Exploring by the Seat of Your Pants broadcast. Now, I know we've got some familiar faces in the crowd, but our speakers are new to us too. So I will note that we are all about bringing conservation, adventure, and science into classrooms around the world. Now, everything we do goes through our YouTube channel. So if you want to share this one in 3 weeks, in 3 years, if you want to catch any of our other amazing Gairdner Foundation series, we have been partnering with them all year long for some really amazing broadcasts featuring their laureates, some of the most incredible biomedical and health researchers on planet Earth. It has been a wild ride. We've done everything from CRISPR to cancer, maternal health, and more. Again, all on our YouTube channel. Can't encourage you to check it all out enough. Now, today, I'm really excited because we're welcoming in 2 researchers, a seasoned researcher and an undergrad in his first year, to talk about some really amazing work in bioinformatics and the platinum age of virus discovery. So we all just lived through a pandemic. It was a long 3 years. We're still in the middle of it, actually. COVID has not gone anywhere. It's still around. Half my people in my neighborhood have COVID right now. So to prevent our future pandemics, to try and understand more about how these come to be and what we can do to stop them, we need to start with virus discovery, understanding what's out there to begin with. Now, our researchers du jour have done more to do that than pretty much anyone on planet Earth. Artem, joining us in just a second, is the head of the RNA lab. Really, really cool science. I'll make sure to link that into all of our classes when we're done this. But using a mix of cloud computing, AlphaFold, which is an amazing tool, AI, and more, they're seeking to understand more about the viruses that are all around us. So without further ado, I'm going to bring in our researchers. Thank you so much for joining us today. Artem, welcome to the broadcast, man. And Rob, nice to meet you.
Thank you for having us, Jesse. It's a real pleasure to be here.
So as I said, we've got a lot of classes. I know you've got a lot to share with us today, and I really want to have a nice big Q&A at the end with some really viral keen kids, as it were. So if you want to dive in and show us about some of the cool work you're up to, let's dive in, man.
Yeah, happy to do so. Just give me 1 second. Let's share the screen. And is that coming across okay?
That is perfect. Let's do it.
Okay, so hello everyone. My name is Artem Babian. I'm an assistant professor here at the University of Toronto. I actually just opened my lab a year ago, so we're all fresh-faced here. And the main focus of my lab is on RNA virus discovery using large-scale computing and things like cloud computing. So the title of the talk today and the journey I'm kind of hoping to lead you into is Entering the Platinum Age of Virus Discovery, or why right now is probably the most interesting time to be in the field of virus discovery ever. So this project, like many virology projects, began at the very beginning of the pandemic. So if you kind of think back to March of 2020, it was really obvious to me, and I was just a postdoc researcher, so I just got my PhD. It was really obvious that this is going to be a really big society affecting event. And, I'm talking about COVID-19, of course. And I saw how all these researchers around the world were spending billions of dollars monitoring how this virus was evolving and spreading across the world in essentially real time. And I felt, as a researcher, I wasn't directly working in virology, but I knew kind of how to operate in that space. I felt it was my responsibility to kind of do my part, find some kind of research question where I can help fight or mitigate this oncoming pandemic. And so I decided to ask a bit of a different question, not so much where is this virus going in real time, but where did this virus come from in evolutionary time? What is the origins of SARS-CoV-2? And so SARS-CoV-2 is one coronavirus from the family, from the subfamily beta coronaviruses. And there's 44 known species of coronaviruses when we began this work. And so this wasn't just an academic problem. There was a concern I had at the time that if SARS-CoV-2 infects another species of mammal, say cows or dolphins or sheep, it was predicted that it could infect over 80 species of mammal. The concern was that when it entered one of those other mammals, it would change in those mammals. And then if that were to go back into humans, a spillback event, that would actually present a danger. So as we were vaccinating or developing herd immunity, these changed viruses, when they spill back, they can essentially escape that herd immunity, meaning that it would be like the entire pandemic restarts. That was actually quite prophetic. So SARS-CoV-2 has now been reported to infect over 24 species of mammal, including stable infection of white-tailed deer in Ontario and Michigan, where a earlier variant of SARS-CoV-2 is spreading between deer and is likely to exist in that animal population for in perpetuity. And so this idea that, the health of humanity is completely interlinked with the health of the animals surrounding us. We can't just study viruses in isolation as human pathogens, because we now have a case study how a bat coronavirus in China causes trillions of dollars of economic damage in North America just a year later. And so this idea that we have to understand the totality of viruses or different pathogens in all the different animal reservoirs and all their different environments is called One Health. So it's kind of a holistic understanding of viruses. And that's kind of what we're working towards, is not just understanding SARS-CoV-2 in isolation, but kind of seeing the big picture. So I came up with this idea that we would be able to find novel coronaviruses in data that was generated by other researchers. So what we were looking at was DNA or RNA sequencing data. So when researchers go all over the world and they're doing their research, they can generate DNA or RNA sequencing data as part of their normal experiments. Think of it as like if I'm researching cows and I'm researching how their gut digests their food, I would sometimes do RNA sequencing on their gut and then look at what kind of RNA is there. And if I'm not interested in viruses, I would never look for viruses per se. And so that type of data has been accumulating exponentially for the last 15 years, meaning that there's millions of samples that are available in entirely public and free databases. This is an enormous volume of data. If you were to quantify how much data that is, this is something on the order of 100 million gigabytes of data. If you were to quantify what the cost of that data would be to run the machines to make that amount of data, is on the order of 3.7 to $14.9 billion. So from my perspective, I'm just was a sole researcher, but I have access to billions of dollars of publicly available data, which I could use to answer my question of, are there missing coronaviruses? And so, the way that I'm starting to think about this, and this is actually where I think computational biology is really strong, in that there is a strong ethic of data sharing. And so we have access to billions of dollars of data. And this represents things like tumor samples from Sick Kids Hospital in Ontario. There's ant mound sequencing from a mound in the Amazon basin, so soil sequencing. There's even anal swabs of penguins taken in Antarctica and everything in between. And all of that data is sitting there publicly available for us to analyze. And so, I won't go into too much of the technical details, but we built this cloud computing architecture called Ceratus that we could analyze this enormous volume of data very efficiently. We got it to the point where we can analyze over 1 million data sets per day, and the cost to analyze a single data set was under 1/2 cent per data set. So meaning, for the first time, we could analyze the totality of sequencing data produced all over the planet faster than the world is producing that data to begin with. And so, when you build a really, really big hammer, everything starts to look like a big nail. We went and decided that we weren't just going to look for coronaviruses, we were going to look for all RNA viruses. So things like Ebola or rabies or influenza, and in all the different species that are out there, in plants, in insects, in fungi. So we went to public databases. So these are where known sequences are stored. And we collected and curated all known RNA viruses at the time. And that represented 15,000 distinct species. And we use this one gene called the RNA-dependent RNA polymerase. You can think of this as kind of the beating heart of an RNA virus genome. By definition, all RNA viruses must contain this RDRP gene. And so if we're able to analyze and barcode this RDRP gene, we could essentially find out where these RNA viruses are across that entire sequencing data set. And so using Ceratus and using this RDRP classification system, we searched over 5.7 million sequencing data sets, or 10.2 petabases, so that's millions of gigabases. So you can think of that as like millions of gigabytes of sequencing data from all over the planet. And we discovered over 130,000 new species of RNA viruses in a single 11-day analysis. So to put that into perspective, it took all the virologists in the world over 130 years to discover the first 15,000 RNA viruses. But by using computational biology, we were able to find the next 130,000 RNA viruses in under 2 weeks. And so this is now turning what used to be a very complex computational problem into something that we can essentially just go to our web browser where we're serving all of this data, and you can just look up and say, okay, I want to find novel coronaviruses. You just select coronaviruses that are under 90% identity to a reference sequence, and it'll tell you where the novel coronaviruses are. And that's essentially where we landed. The beauty of doing a big unbiased screen is you find things that are really unexpected. And so we found new species of coronaviruses in things in aquatic vertebrates. So the axolotl, a seahorse, a fugu fish. This is not where I would normally consider where I would look for coronaviruses, but that's where the data led us. And so that's where we went. And then when we went into characterizing what the genomes of these new coronaviruses looks like, we ran into a problem where we were able to find that RDRP gene that we were looking for, but all of the structural genes, the things that make up the physical viral particle, they weren't found on the genomes. And after a little bit of sleuthing, what we actually identified was that the RDRP and the non-structural genes occurred on one molecule of RNA, and the structural genes occurred on a separate molecule of RNA. And so in terms of RNA viruses, what this means is this is now a segmented virus where instead of one molecule, the genome is represented on two molecules. So classically, you think of viruses like influenza as being segmented. But here, for the first time, we were able to show that this type of coronaviruses have segmented genomes. And if you open up your textbook, it will say that all coronaviruses must have one molecule. And here we have a clear violation of what the textbook definition is of a coronavirus. This is where I think the most exciting type of research can happen when you do this computational discoveries, is you're going to find things that don't make sense with the textbook definition of what something should or should not be. You're making real, honest discoveries every time you're going about and doing this research. And this actually fits into a much bigger framework. So the World Health Organization put out this white paper, this guideline for how genomic surveillance has to become a bigger part of what we do, what the world does in terms of public health for monitoring things like future pandemics. And one thing that the World Health Organization highlights is that computational analysis, it comes with a high technical and financial cost. And so I view kind of the mandate of our research lab here in Toronto as overcoming what that high technical and financial cost is and being able to distribute world-class analysis tools into resource-limited settings, right? Our job is to build these analysis platforms and then deliver it so that people in the global south, say, have access to state-of-the-art analysis techniques. And this kind of comes into a much bigger picture. We're entering a world where there is going to be an enormous amount of data. And we're all inundated by this huge volume of data. But that data is meaningless unless it can be converted into meaningful knowledge. So knowledge, I mean things like a Wikipedia page or a figure or a scientific publication or an infographic, somehow to make sense of this big, massive data. Because by creating human interfacing knowledge systems, that will give us wisdom. And that means the ability to make the correct decision. So when the next pandemic rolls around, we can act more wisely. We can act to save more lives in a evidence-based manner. And so, again, scaling back now, the way we're looking at this problem moving forward is that as the entire population of biologists over the world are producing more and more sequencing data, we can analyze the totality of that data and create a kind of passive planetary-wide viral surveillance network. So the idea is if another pandemic agent shows up, we can identify it sooner, so where we can act and make an intervention that will be more meaningful to not have another COVID-19 event happen. And so you can ask, okay, are we going to find another 150,000 viruses in a year because the data is growing exponentially? Well, no, that actually is an underestimate. But I think this ties to kind of the most important point of the research program to begin with. This project, Ceratus, and what our lab is doing, it's not really about patting ourselves on the back and saying, okay, we took the known virome and we blew it up by 10-fold, good job on us. I think the most exciting part of this research is if you put that into perspective with the known biodiversity of viruses that are out there in nature, we have not even scratched the surface. There is a huge repertoire of viruses out there, and we have no idea what they are. They're going to have completely new types of biology. And so, the job of our lab now is to ask questions like, okay, which virus is potentially causing cancer? Or which virus could cause a disease like Alzheimer's, where we don't know what the cause of Alzheimer's are, right? So by doing this, we can make clinically and human relevant discoveries that we're able to share with the world. And so if you combine this growth of data production and increased capacity for data analysis, we are on track to finding over 100 million new species of RNA viruses by 2030. And more importantly, it's not just going to be about finding more and more viruses, it's going to be about asking which one is the next one. So which one is going to cause the next pandemic, or which one is causing Alzheimer's disease? By answering that question, we'll be able to do kind of more significant research that will impact people's lives on a day-to-day basis. So with that, I'm going to turn it over to Parav. So Parav is he started as a high school student here. He emailed me and was interested in doing virus discovery. And so I gave him a problem and he just took to it. And so while he was still in high school, he made the discovery that he's going to be telling you about. And then we turned that into now a scientific publication, which we're working on. So from there, I'll pass it over to Parav.
Great. Hi, guys. My name is Parav. And today I'll be talking to you about a summer project that I worked on, and also how I really got into this field, what I did to get into this, and then there's a bit about my story, and then hopefully that will spark something in you. All right, so to start off, a parasite odyssey, an RNA virus concealed in toxoplasma. So you may be thinking, what is toxoplasma? It's just a big word that I just spewed at you. But toxoplasma is actually a global pathogen. It is extremely prevalent in the world, and it is a cause of toxoplasmosis, a pretty prevalent disease in many parts of the world. Now, it has the ability to infect all warm-blooded animals present, with about more than 50% of the world's population being infected by this parasite. And just some other facts about it, toxoplasma has a unique ability to move up the food chain, and in doing so, it has a unique ability to, really spread itself throughout the whole population. So, why are we looking at toxoplasma, and what is our main goal that we tried to answer in our lab? So Artem mentioned something about Alzheimer's, and Alzheimer's is a unique disease which has hallmark brain inflammation. Now, our hypothesis was that there might be a virus, an RNA virus, that infects the brain and can lead to this brain inflammation, therefore leading to Alzheimer's. So the dream would be to find a virus that causes Alzheimer's and then maybe make a vaccine to prevent the onset of Alzheimer's in the future. So that was our dream and our goal, and we worked towards that by making a hypothesis that there is an RNA virus present in the brain that causes Alzheimer's. So we screened the whole Ceratus database, and we tried to look for this virus. And that leads into my summer project. Now this here is a study that was conducted in 2017 by No et al. And they infected 3 human cell lines with 6 different types of the Toxoplasma gondii parasite. And that led to a whole bunch of data that we analyzed. And upon analysis, we basically found that there were 14 different test samples, and amongst that, there were a bunch of different samples. And one specific strain of the Toxoplasma gondii parasite, called the ruby strain, we found our virus. we made a hypothesis that, all right, if we have a virus present in one strain of Ruby and it's present in one line of neuronal tissue, is it going to be present in other, in other brain tissue? And evidently enough, it was. And that was a good indicator that this virus is present in a good amount of brain tissue, and it is only associating with this one strain of Toxoplasma called Ruby. Going forward with that, we did a whole bunch of other analysis over the summer, and I made this figure here, which really shows that if you look at this right-hand side column, which has the basically the red column, that shows you that our virus is only present in an atypical Ruby strain, which we call the hypervirulent strain. And we basically tried to show, or I tried to show that this virus is sort of present in T. gondii, like a Trojan horse. And I'll be elaborating on that a bit further. All right, so where else does our virus appear in, the whole database? We requeried our virus against the whole database of all different viruses, and we found that among 7.5 million sequencing runs, only one project had our virus present in it, and that was this other paper published by Milo in 2013. We found our virus in another Ruby strain of T. gondii, but interestingly enough, we found our virus in another strain, the Cougar strain. Both of these strains are hypervirulent, and they both have our virus present in them. In their study, none of the other 28 strains of T. gondii had our virus present. So our virus is present in only two strains, that are the two hypervirulent strains called Cougar and Ruby. Now, as I was mentioning further, they're hypervirulent, and they are pretty evolutionarily distinct. So if you can see on this phylogenetic tree here, Ruby is pretty far from Cougar, and that basically shows that they are pretty evolutionarily distant, and it is interesting that a virus can infect both of these strains, which are really far apart. So that really ties into how our virus, came to be. The study that showed the Cougar, basically that showed from the Milo 2013 study, was talking about a data set from 1994 of a toxo outbreak in Victoria, BC. They referenced this study about the outbreak in Victoria, and they showed that a cougar was infected with this parasite, and it led to a whole lot of other Toxoplasma infections in humans as it infected the water body present. Right. So going further into their paper, the Milo 2013 paper, we really looked further into their data set and tried to figure out why are only a few strains of Toxoplasma gondii infected with our virus? What about the other strains? And looking more into the data, the Milo 2013, like those researchers also had similar, thought processes, and they, in their paper, wrote that these atypical strains were capable of inducing high levels of expression, the most potent being Ruby and Cougar. And there's some small snippet over here at the bottom that we can recognize bacteria and viruses, and there's an antiviral response going on when they conducted their study. So these really were some good indicators that something unique was going on, and that our virus is, really causing an immune response in the brain. So we did a whole bunch of analysis, and over here we can see that this is basically a differential gene expression of what is going on in the human brain. And the point of note is that on the top right-hand column, you can see that there is a cellular response to a virus if we model it in human tissue. So the Ruby strain is causing a cellular response to the virus. And all of this comes together to really show that in Toxoplasma gondii strains that do have our virus, they are leading to a huge immune response in the brain, versus other T. gondii strains that don't have our virus, they lead to typical asymptomatic infections. So where does this leave us? It left us with 18 new RNA viruses across 195 samples, and we dubbed this the K virus Odysseus type viruses, and they are closely related to another family of viruses called narnoviruses. And this was a whole family of viruses that we found from these studies. Okay, so the whole thing about the study is that we tried to look for a new virus, trying to find trying to maybe cure Alzheimer's in the future. We tried to look for, we had a goal, and we found something which is completely unexpected. So that's the whole goal of this lab, that we try to expect something, but we always get something unexpected. And that was the whole summer project that I worked on. Now, you may be thinking to yourself, how is this, really tying into a high school student coming into the lab? Now, as you can see here, this is everybody in our lab, and most of the people here were at least 3 to 4 years older than me. So how did it go from me being a high school student to getting into this lab? How did I get here? Now, the next part of this talk may be a bit fun for you, so hopefully you'll enjoy some Kung Fu Panda analogies. How did Po was chosen by Master Oogway? How was he chosen as a dragon warrior? It was more so because Oogway could see inside Po and could see his inner passion for Kung Fu and could see his inner interest, and that is the key driving factor for everything. So let me ask you a general question. Which type of student are you? Are you on the left here? Are you Po, who is bored in some of your classes, and you're not necessarily bored in every class, but you just have a genuine interest to some subjects, and in other subjects, you continue on with the work, but it's not your true passion? Or are you like this Po? Are you like this Po who truly loves the stuff they're working on in class? And for me, that was math, bio, chem. So are you like him who has an idea of what you like? If you are, then that's amazing. How do you go from that to breaking outside of your shell and going across the curtain into the real world? Well, for me at least, it was cold emailing. So what is cold emailing? Cold emailing is basically what I did to get my this internship, or to get this undergrad position. I sent about 30 to 40 emails to professors at both UFT and McMaster, two universities in Canada. And I basically went to their website, I looked at the faculty list, and just sent an email showing my genuine interest in their work, and I gave a good background of who I am. And I sent these emails to different professors at these two universities, and one did get back. A couple got back. So yeah, cold emailing is how it really works in the real world. When you come to university, or when you go into any type of position, looking for an internship, a job, or anything, your network is the key factor. And if you can, network with people, and if you can sort of get this opportunity early on, it would be great. Okay, so let's go through this timeline. In grade 12, early on in grade 12, I sent out a bunch of emails to the different professors at these universities. And I luckily got a position, which I started working on with Artem's lab from latest December. My early work was literally me trying to learn the basic skills, because I know all of you high students out there, we think that our whole life is just school, work, university applications, but there's so much other cool stuff happening in the world. So how do we go from just learning about biology in class to really doing these cool projects in the world? And that's what I did. I spent about 3 to 4 months on, and I really tried to learn the different basic skills that are required for this field. If you can see here on the left, there's this rotating image. It is of the RDRP that Artem talked about. It is the beating heart of an RNA virus. This, I created this on AlphaFold, and I sort of labeled a couple of the key structures in it. And the point that I want to drive home is that all this analysis was done on Google Colab for free. All of you, everybody with a Google account, has access to a virtual machine which can run these crazy simulations and can run these crazy programs. So sitting from the comfort of your home, you can do all of this just with the access of one laptop. Right. So this was some of the work that I did over the summer. This was a paper that I published called The Parasite Odyssey. And so how did I get from the early work to doing this? It took over a couple of months over the summer, and just some genuine interest into doing the things that I liked. And with a bit of belief, I bet that you can make these type of figures too. All you needed is a laptop and just a drive to do this. Okay, so let's go back to Po. When Po was a dragon warrior and he got the dragon scroll, he thought that he was getting the secret ingredient, the dragon scroll. Now, as some of you may have seen the movie, you know what is coming, but in our initial days, we may think that there is some type of secret ingredient, like if we go to university and spend 4 years, we'll magically get into the real world and then be able to start solving some crazy problems. But as Po learned, there is no secret ingredient. It's just you and the way that you want to approach a problem, the way that you truly are genuinely interested in something. It's all about the initial spark. And hopefully giving you these analogies can give you that spark and can show you the other side of a curtain. That it's not just high school. There is a whole world out there in which you as high school students can easily get into this field and can easily do some crazy discoveries. Now, let me run through an example with you. Let's say you are the student who is just, doing good in school and who is, really liking their time. They are having fun, going through your courses, they're having fun with friends, and, they're just like going through the motions of life. How do you go from this student to being somebody who can, cold email people, who can go into then research? All it takes is that initial spark. With that initial spark and with that initial genuine interest in something, you can go from being just every single high school student to being a couple of them who want to do something in the world. And luckily enough, in today's day and age, all you need is a laptop. With a laptop, you have access to the whole world at your fingertips. Now, in my case, with a laptop, it gave me the power to discover new viruses. I could discover a new virus sitting in my science classroom in grade 12, just working with my own laptop. And in doing so, I discovered a new virus, and that opens up the possibility to the whole world. You have access to professors right at your fingertips. You have access to the whole SRA, the whole billions of petabytes of data right at your fingertips. You have access to infinitely amount of tools like assembly, whatever, AlphaFold. All of that is from your laptop. So the key point that I'm trying to drive is everything is accessible to you. Everything can be done right from your laptop. All it takes is that initial spark. For me, it was through actually one of these talks or one of these presentations that I attended over summer. Over summer, I learned that research is not a daunting field. Research, or rather any internship, any real life opportunity can easily be acquired if you truly look for it. And in doing so, I then, going in my grade 12 year, I emailed people and I got a position. So I hope that really showed you that research, or anything for the matter outside of school or anything outside of your normal way of thinking right now, is extremely accessible and possible. I wanted to give a huge thank you to everybody in our lab who made all of this possible, and to Artem here, of course, who made everything that I did possible and for accepting me into the lab. So I really hope that today you got that spark, which I got, to maybe go out and email the next professor, to go out and find the next internship, or to create your own project for that matter. I really hope this sets the ball rolling for the future. Thank you.
Fantastic, gentlemen. Well, we've covered a lot. I really, if you'd asked me at the beginning of my career whether I thought I'd have a broadcast with dark RNA virus, Kung Fu Panda stuff, and anal swab of penguins in one go, I really would have said that would never happen. So thank you for crossing that off my bucket list, you two. Guys, we're going to dive in with questions. Is there anything else you wanted to share before we start taking questions from our audience? Or
no, I will just add one thing. If you're going to be doing cold emailing, the most important thing is to make it specific. So as opposed to just sending off a bunch of emails, it is not a numbers game. It is about finding the few researchers that you really are genuinely interested in their research program, and making that email specific, saying I really am interested in this topic, and here's some evidence to demonstrate my interest. So being able to do that, because otherwise we get tons of emails and it's hard to resolve people who are genuinely interested versus people who are sending off hundreds of emails, which does happen too. So specificity, quality over quantity on the emailing.
Yeah, this is something that, it's funny that you gave such a concrete example of this in the high school context coming into undergrad, but we tell our classrooms all the time, scientists don't bite. Most scientists are really keen to share their work, to have people involved, to whether it's a lab tour or highlight or answer some questions. It's quite amazing how far cold emailing can go. My entire career is based off cold emailing. So I'm really glad we talked about that in a big way today. Guys, we're going to dive in with questions. YouTube audience, we'd love to hear some questions from you, Mr. Gerard's class. I'll be coming to you in a moment live, but we're going to dive in with one from YouTube. So Ms. McCool's class, welcome in, folks. They want to start off with, do you think scientists are positive in their classification of viruses? So how do we know that it is what it is? However you want to interpret that, I'll leave it to you.
Well, that's actually a fantastic question, because any of these classifications of viruses, there is an organization called the ICTV, and they're very slow in kind of confirming, and they have panels. So when it comes to viruses that are, say, clinically relevant, like cold viruses, HIV, or SARS-CoV-2, these panels essentially decide what is or is not part of a classification. When you're on the research side, it's kind of the Wild West, right? If we discovered, like, as one research project, we discovered 10 times more viruses than everyone in the world combined, we can't make official classifications. That's 10 times more work than everyone. So it really comes down to at what level of analysis, right? It's kind of when you're doing research, the boundaries are a little bit more loose. When it's going to things that are clinically relevant, you have to kind of have committees that come and classify viruses officially.
Thank you for the nuanced answer. By the way, the 10 times thing is really wild. I think some of our audience might be familiar with this, but most advances in science are like 1% better. And that's thrilling. Like, I mean, if you can do 1% better in anything in science, you're like over the moon. 10 times is unheard of. So I really appreciate we got a unique story in that way today. We got a great question online. So this is from one of our email classes in. We do a lot of eDNA work talking about this in our broadcast, Exploring by the Seat of Your Pants. So expeditions go out, run something through the water, collect things for sample to get eDNA samples. Would an expedition type thing work for the work that you're doing, or is it moot point in comparison to the reams of data that are out there? Like, would there be a benefit to something like that for the RNA viruses or not?
Well, I mean, that's a fantastic question. Most of the viruses that we discovered are from different types of environmental DNA. So there's kind of a trade-off. When you find a virus in an environmental sample like soil or water or air, you don't actually know, like, what is it infecting. You just know roughly its geography or what kind of environment it's in, which could still be very important information because, it's important to know which regions of the world are different coronaviruses found in, for instance. But from our perspective, it doesn't make sense to go and invest resources into trying to generate more data, because my philosophy is that there's already billions of dollars being spent on generating more and more data, and those people will make that data. My job is to come in and do a secondary analysis on that for a pittance of the cost, right? The entire Ceratus project, that like 10-fold more viruses than everyone in the world, that costs $24,000, right? Which is a lot of money. But in terms of research, that essentially is nothing. We are competing with these kind of expedition type projects where their budget is in the billions of dollars, and we're talking about tens of thousands of dollars of computing.
Well, that is incredible. $24,000 for context. I mean, for our students, we get questions about the cost of expeditions all the time. It's the smallest volume I've ever actually heard of for anything. I will say, however, that you two would look really cool on one of those research ships, though. Like, if you want to, and you can do the computing in the bottom, I'll do a plug-in for you. We're going to head to Notre Dame High School, Belmont. If you guys want to come on in with a question, we'll head to you, and we'll try and take as many as we can live from this class over the next few minutes.
Yep.
Hey, ladies.
Annette, and then, sorry. Okay. Yep. Hi, I'm Annette. I was just wondering, because you talked about how you discovered there are new species of coronaviruses in aquatic species, like how the coronavirus would affect those species, like axolotls.
Interesting.
Well, if you think about it, we're all aquatic on a long enough timeline, right? Life originated in the oceans, and the biodiversity of the oceans is actually probably greater than the biodiversity of like land animals. We're, one way to think about it is we've encapsulated our water and we've moved it onto land, but we're still mostly water. So in the oceans, that boundary of like organismal self versus non-self is a bit looser. And so like if you're sloughing off, in the same way that we have like droplets in the air, you just get droplets and particles spreading in the ocean. For the case of the axolotl, though, those were not wild axolotls. Those were in laboratory animals. So this is what is kind of meant by incidental findings. The people who were studying those axolotls were studying limb regeneration. So they were cutting the limbs, and axolotls have this ability to regrow their entire limb. And so they were doing RNA sequencing on limb regeneration to better understand that process. They had no idea that their animals had this coronavirus in them, but the sequencing data was able to pick it up. And so they deposited that data. And then when we went back and analyzed it from those lab animals, we were able to identify that coronavirus there.
Very cool. I will say, and you can correct me if I'm wrong, but it tends to be the case that if something really affects one kind of species, it's most likely to have a significant impact on other close relatives. So like chimp virus would be something that would affect us, and that's what we have with HIV. That's what we have with Ebola, with gorillas, same sort of deal. And the further you sort of get in the evolutionary tree away from that, the less likely it is to have a sort of negative big impact. It can, but I don't know a specific example of that, just broadly speaking with this question. Can you?
I mean, of course, there are exceptions. It's biology. Yes. It wouldn't be biology without exceptions, right? So yeah, the axolotl and the fugu fish, right? Like those viruses are more closely related to one another, and they will not infect humans. There's zero evidence that they would have any pathology. Most viruses won't infect humans, right? So viruses are usually quite specific in what they can infect. But the exceptions are things that are like Zika virus, for example, can infect mosquitoes, which are insects, and it can infect mammals, which is like, if you think about it, that's a wild host range.
That is. I'm just bringing up a banner for a book. If you are curious about zoonotic viruses in general, Spillover by David Quammen is still like the best popular science book ever on that. It's an amazing read. He did a follow-up after COVID too, but that's a really incredible book if you want to keep that learning going. Ms. Gerard, I'm going to come back to you live because you do have a bunch of questions. We've got about 6-7 minutes left, and we've got some more on YouTube too, which is great. But Notre Dame, come on back in. Hey.
Hi, I'm Tori, and I was just wondering that, like, when you find like thousands of like new RNA viruses, how do you identify which ones like could cause a possible pandemic or like cause something specific like Alzheimer's?
Great question, Tori.
Do you want to take it? How do we do it?
All right. So, well, at least for the Alzheimer's case, how we really did is we really screened for like to see if they are present in brain tissue, and if they are, then we did a whole process of going through and finding and figuring out what the virus actually does, and seeing what type of cells it can infect, what are its pathways that's infecting, and what's the whole process of the virus present in the brain. And then that is how we do it for Alzheimer's. And then for the whole pandemic, also, we can really look into what are the different types of animals that it infects, and to see if this virus can infect closely related species to humans. As Jesse was mentioning, actually spillover. If it can infect, then it could potentially have some pandemic qualities, and then then we go from there to see what it truly is.
There is no good answer to this question. It is like many research labs all over the world are working on this, because if we can identify those, what is the properties that makes a pandemic virus pandemic, then we can prioritize. And that's actually really important. So a huge part of the lab now is being able to prioritize the RNA viruses.
I'm so glad you highlighted that. Like, this is one of the most exciting times in virus discovery and seeking to thwart pandemics. I mean, like, literally strike while the iron's hot. We all lived through a once-in-a-century pandemic that might be likely to happen more than once a century, given our global travel right now. So it's a really exciting time to be interested in this field. So Tori, our whole class, anyone who's tuning in, like, if you're keen on this, get involved now. Look into that research. Do cool emails like Parav. Like, I mean, it's thrilling, honestly. Take one from YouTube, and then I'm going to head back. Ms. McCool asked a question that we've gotten in every one of our Gairdner programs. Do you think AI will be beneficial to scientists? Can it save time? And in the future, will it take over in some fashion in terms of the research that you're doing? AI goes we can take that a lot of places, but specific to your research, how helpful is it? Where are we going in the future with it?
Yeah, I mean, we're using AI all the time in the lab now, right? We're using it AlphaFold. So we actually invested a lot of resources into predicting structures, because that's a different way where we can identify this RDRP gene is through structural space. And we're also using it for things like trying to interpret scientific abstracts or metadata, being able to make sense of like the large volumes of metadata that is out there that's also associated with these sequencing data sets. So there's a huge future for AI, certainly. Will it take over? I mean, probably. It's probably a good time to learn to maintain the AI, because when the robot overlords take over, then you'll be useful to keep you around.
There's your plug of the day, kids. We're going to take a couple more questions before we wrap up. Notre Dame, I want to come to you one more time, as you guys have the best questions every time. So come join us again and take us away. Hey.
Go for it.
Hi, my name's Savannah. I was wondering about the Ceratus architecture. How was it developed? Was it coding? Is it like computer software?
Cool.
Yeah, I mean, it really is just some kind of computer software. It was me and my climbing partner, Bree Taylor. We were just talking about like, oh, we think we can do this cool analysis as we're climbing. We went back to my place, sketched it out on a napkin, and then we just spent over the next month developing how we would use cloud computing. And it was really just this idea of like, we could, instead of trying to do really big analysis, could we break up the problem into like microservices and run very small analyses, and then run a distributed system that way. And so that idea, it's called horizontal scaling. If you're able to do it with this type, like this class of problem, which are called parallel problems, then it actually works very efficiently. So we built that over about a period of about 3 weeks. We got a prototype working, and then we spent another about 2 months optimizing it. And then, that's essentially it. Once we built the tool, the world was our oyster.
Yep. That actually was pretty much exactly the question that I was going to take from online. So if we want to do one more and then we'll wrap up from there, I do want to note you highlighted a preprint of yours that I'll share with our classes at the end. So if you want to check out some of the latest and greatest research from Artem's team, please do check that out. RNAlab.ca has so much more. So if you want to discover more about the lab, you want to reach out, if you want to cold email them with some of those specific questions, you can do that right there. A lot of fun. But because you've been so great, YouTubers, if you guys have any additional questions, please do message me as well. It's been a great audience today. But Notre Dame, if you have a final question, we will come to you live. I'd love to hear from you one more time if you're keen.
Hi, I'm Megan. Is this a public website or database that other scientists then can also use in their research?
Yeah, absolutely.
Are people using it?
Sorry, what was the second part of the question?
Oh, sorry. And are there already people going into the viruses that you found with your?
Yeah, that's a great last question. So yes, we use publicly available data, and kind of like the ethics of the Human Genome Project is that DNA and RNA sequencing is a heritage of humanity, and it should be publicly available to everyone. We take on that philosophy as well in the lab, and so all the data that we produce, we release it into the public domain. So we have no copyright, no ownership status at all of any of the data we make. It's just out there as a public good, and people do use it. So 6 months after the Stratus paper went online, a group in Stanford identified that a type of herpes virus reactivates in a cancer therapy, a cancer cell therapy. So the short story there is they were able to discover that if you take T cells out of a cancer patient and grow them in a lab, it causes a reactivation of this herpes virus, which means that you are then putting those T cells back into a cancer patient. And that, they believe, is leading to a side effect of this cell therapy, which was encephalitis. So for me, the entire vindication of the project was that 6 months after we put our work online and made it available, it had a direct application into an FDA-approved cancer therapy.
Wild. Science rocks. You guys are great. Thank you so much for all this, your enthusiasm, your passion. Again, I really encourage everyone to check out rnalab.ca. Parag, you talked about toxoplasma, and I know you talked about it from your context. Everyone should look up toxoplasma. What it causes mice to do and their attraction to cat urine after that is just like black magic. I'm convinced it's half the reason that there's people with cat hoarders around the world. So some really, really cool places to take this up. I've just emailed some book recommendations to our classes as well to keep the learning going. But gentlemen, this has been a lot of fun. I'm going to bring in Mr. Gerard's class on YouTube. If you guys want to say a big thank you and farewell, even in your home as well, come on into Notre Dame High School. Thank you so much, everyone. Have a wonderful day. Bye for now.